Hello, fellow neuroscience enthusiasts. Today we're going to talk a little bit about chronic pain, in particular, allodynia. Allodynia is when something that doesn't normally hurt causes pain. An example of this is when you have a sunburn and the slightest touch is extremely painful. Now, before I move on, I want to clarify that this is distinct from hyperalgesia, another characteristic of chronic pain. Hyperalgesia is when a normally painful stimulus hurts even more. Stubbing your toe usually hurts, but it hurts even more when you have injured your foot previously. Despite often being used interchangeably, allodynia and hyperalgesia are mediated by different pathways. To understand how allodynia occurs, you need to be familiar with labeled line coding. This theory states that there is one pathway for touch and a completely separate pathway for pain. This is mostly accurate, except the touch pathway has an inhibitory connection to the pain pathway. When you think about it, this actually makes sense. Imagine you are using a hammer to drive nails. Suddenly, your hand slips and you hit your thumb. What do you do next? If you're like most people, you'll hold your thumb or even put it in your mouth. This is because the sense of touch inhibits the pain signals. Taking a closer look at this connection, we can see it is mediated by a GABAergic interneuron. You may recall that GABA is usually an inhibitory neurotransmitter. In a normal state, this might make sense, but what about a chronic pain state? In allodynia, we know that touch actually stimulates pain, so how does this connection flip from being inhibitory to excitatory? It is important to remember that a channel causes excitation or inhibition because of the relative ion concentrations. In most situations, the extracellular chloride levels are much higher, so when GABA channels open, these negative ions rush in and hyperpolarize the cell. However, that is not the case with the nociceptors in the spinal cord. These neurons have higher intracellular chloride, so opening GABA channels is actually depolarizing. You may be wondering, how would a depolarizing current cause inhibition in the first place? Under normal circumstances, the depolarization caused by GABA isn't enough to cross threshold, but is just enough to inactivate voltage-gated sodium channels, preventing an action potential. This is called primary afferent depolarization. In a chronic pain state, there is a much higher concentration of intracellular chloride than normal. When the channels open, even more chloride ions rush out, resulting in greater depolarization. This depolarization is called a dorsal root reflex and causes an action potential. Now, touch is perceived as painful. For this reason, the relative concentrations of chloride are extremely important. The concentrations of chloride are controlled by the activity of transporters in the membrane. A team of researchers in Quebec hypothesized that targeting these transporters could be an effective pain therapy. They began by developing a drug to selectively enhance KCC2, since this transporter is only expressed in the central nervous system. Starting from a library of 92,500 compounds, they narrowed it down to one drug, CL58, and then began the optimization process. Researchers synthesized over 300 different analogs of the original compound. They tested each compound's ability to enhance chloride extrusion using a fluorometric assay they designed themselves. Essentially, they transfected cells with the protein chlomelain, which glows in the presence of chloride ions. The greater the concentration of chloride, the brighter it glows. They chose CLP257 due to its efficacy and stability. However, during testing, they noticed the drug had a short half-life of less than 15 minutes due to metabolism by liver enzymes. They designed a prodrug, CLP290, to be administered instead to improve the half-life. A prodrug is a compound that turns into the active drug after being metabolized. CLP290 is metabolized into CLP257 and allows the drug to stay into the system for a longer amount of time. After assessing its toxicity, the drug CLP290 was ready for animal trials. When administered to normal rats, there was no change in response to painful stimuli. However, when given to rats in a chronic pain state, it negated the sensitizing effects. Rats with the drug had a lesser response than rats given saline. When compared to a common pain reliever, pregabalin, CLP290 was just as effective at relieving pain, while not causing any motor deficits common to pregabalin users. Overall, this drug was quite effective at relieving pain in rats. And, given that the drug was highly specific for its target, and the fact that its target is only expressed in the central nervous system, it produced very few adverse side effects. Additionally, the drug had a very low toxicity. Further testing is necessary to evaluate its efficacy in humans, but this research could potentially lead to a new class of pain relievers. Thank you for watching, and I hope you check out the resources listed